Good afternoon, everyone. Welcome to the seminar, the first seminars of 2026 on Xylella fastidiosa seminar series. Today we have Charlotte Trontin and Rob Turner from EPPO. As you know, Rob Turner presented the simulation exercise at the end of last year. Charlotte and Rob both are scientific officers from EPPO. And I will introduce today Charlotte, that is the new speaker. She joined EPPO in August 2019. And she supports the EPPO panel on diagnostics and coordinates also the preparation of the EPPO Diagnostic Standards. She's also involved in the technical maintenance of two databases on diagnosis. So she also was involved in leading the work package on dissemination and communication and training of the project, European project VALITEST. And also before joining the EPPO, she was working as policy officer from the French National Plant Protection Organization. So thank you, Charlotte and Rob, for being the first ones in starting the seminars in this year. And I will give you the floor. You will be sharing the slides, so just feel free to participate whenever you have planned. Thank you.
Thank you very much, Blanca. Thank you very much, Anna, and thank you very much to the Bexel project for enabling this webinar. And I think I can speak on behalf of Charlotte and myself. EPPO is very happy we're involved in the Bexel project and working alongside the researchers to see what communication work and what guidance we can, what training, as in the simulation exercise, that we can help to deliver as part of the project. So yes, so today this evening, we will be talking about the development of regional standards and how they help safeguard the EPPO region against Xylella fastidiosa. For those of you that saw my presentation last time, I apologize because the first few slides are kind of similar, but I think it's really important to set the scene and to give some information on what EPPO does as an organization and where we're looking to go into the future. So EPPO, the European and Mediterranean Plant Protection Organization, we're an international intergovernmental organization, 52 member countries, and originally created in 1951. This year is our 75th birthday, hence the logo update on the first slide. We're dedicated to the international cooperation in plant health, including both plant quarantine and plant protection products under two working parties. We work for and with the national plant protection organizations of our member countries in the region. The aims of EPPO, I won't read these all out, but I will just highlight the aim is to support member governments ensuring plant health, while particularly also looking at human and animal health and the wider environment, to facilitate and encourage cooperation between our member governments to ensure that we're working towards the common goal for the prevention of plant pests entering the region and the protection of plants within the region. We do this through the development of international harmonized phytosanitary and other official plant protection measures. And we elaborate standards, we develop guidance for utilization within the EPPO region. We have a harmonized development of guidance that can be used across the EPPO region. We work of course, in the international arena, but we also look outside of the EPPO region, and of course for international bodies which also work within the EPPO region as well. So we ensure that we work with other regional plant protection organizations of the globe. We're one of 10 regional plant protection organizations, but we also work with bodies and have strong collaborations with the FAO, IPPC, EFSA, European Commission, etc. I think it's really interesting to highlight at the end, towards the end of last year, 2025, the EPPO Council adopted a new strategic framework for EPPO, short term from 2026 to 2030, which includes 6 main strategic objectives. All of our core work that we already do and have delivered and are known for are woven into these 6 strategic objectives. But then we're giving some more emphasis to enhancing our regional preparedness and response to plant pest threats, engaging and promoting sustainable plant protection strategies, including biological control, strengthening the scientific foundation of scientific phytosanitary regulations through the networking and the utilization of scientific research to be detailed within our standards, strengthening cooperation and knowledge sharing across the EPPO region and beyond. Again, as I mentioned earlier, strengthening and maintaining this global alignment for effective plant health management. And the last one is to really ensure that we have the leadership and the expertise within the Secretariat to ensure that we can continue our role and deliver to our member countries. The EPPO structure. We are the EPPO Secretariat, approximately 20-something staff based in Paris. We work for our national plant protection organizations, and experts from the MPPOs may be involved in expert working groups, ad hoc expert working groups for the production of pest risk analysis, or Pacific panels, whether that's panels on diagnostics of nematology, or whether it's a panel on biological control agents or forestry, quarantine pests for forestry. The panels work to deliver the work of EPPO. I think the most important thing to note from this panel is we're the Secretariat. We really rely on the expertise, the massive network of expertise, and the generous time that they give us to the development of the products, the standards, the information that's included in EPPO Global Database. The working parties are country representation. We have the Working Party on Phytosanitary Regulations. We have the Working Party on Plant Protection Products. We have the Executive Committee that oversees the work, and of course the Council, the member countries that endorse and our standards, our PRAs, but also set the work program for the future years. EPPO activities in phytosanitary measures. This is one slide that encapsulates a hell of a lot, really. We manage early warning systems, for example, the EPPO alert list, where we highlight to member countries of emerging pests. We evaluate risks presented by emerging pests and make recommendations for regulation through the development of pest risk analysis. We maintain lists of EPPO A1 and A2 pests. EPPO A1 pests are pests that are not currently present in the EPPO region compared to A2 pests, which may be present, but with a very limited distribution. We also work on RNQPs, regulated non-quarantine pests. And we make recommendations to prevent the introduction and spread of regulated pests into the region through the development of regional standards. Here is a list of the whole series of different standards that we develop within the EPPO Secretariat. And of course, with this presentation, we'll concentrate on just two phytosanitary inspections and diagnostic protocols, but we deliver and utilize a number of other from pest risk analysis where there's methodologies in the and templates for the assessing the risk of pests to national regulatory control systems, which detail control, containment, and eradication measures for specific pests. Here we have a kind of diagram on the how standards are first of all prepared and the approval process. Initially, the panels and the working parties endorse the preparation of a particular standard. And a drafting team will work on that standard. They may be members of specific panels, but also they may be external experts if we need additional expertise. The panel will develop the standard, and the EPPO Secretariat will facilitate the process. And when the standard is ready for the next step, it would go to country consultation, where our member countries will provide really excellent scientific technical feedback on the contents of the standards, whether that's expertise from MPPOs, but also expert consultation. Following the country consultation stage, the standard may go back to the drafting team and the panel for additional amendments. And when it is eventually finalized, it would go to for inspection standards. It will go to the working party for approval and the council for endorsement. When it goes to the council, then the standard approved. However, there is another little deviation for that with the diagnostic standards, and I'll let Charlotte highlight that to you.
Yes, so for diagnostic, we have a first consultation, which is actually a little bit longer than for the other standards, since it's 60 days instead of 6 weeks. And then instead of being approved and endorsed by the council, the diagnostic standards usually goes through a second country consultation that will only be 1 month. And during this country consultation, it's only to get the opportunities to the countries to make a formal objection on the standard. And if no objection is received, then we consider that the standard is approved. So it doesn't go to the working party and the council. It allows to be sometimes a little bit quicker on the approval of these standards that are very technical.
Thanks, Charlotte, for that. That's really good. Yeah, and I think the take-home message from this slide is that there are a number of different steps in the development approval of the technical standards. So I'll talk to you about the inspection standards that fall under the series phytosanitary procedures. They all have a PM3 number associated with them, and they're intended to be used by national plant protection organizations in their capacities as bodies responsible for the inspection and testing of plants and plant products moving in trade, or for the implementation of surveys against quarantine pests. We have two types of PM3 standards, I think is good to say. We have two more generic overarching standards that can be used in association and consultation with the more pest or commodity specific PM3 standards. And we'll have a look at those ones in a minute. But these are the two overarching standards that give general information which is useful, common for the PM372, common to inspection, places of production, area-wide surveillance, inspection of consignments, and lot identification. Within the presentation, you're going to see a lot of PM numbers, and you're going to see numbers in brackets. And I think it's really important to highlight the numbers in brackets. The number in brackets mean it's the revision number. So for the PM372 here, this is the second revision, and it's under revision again. I think the key point there is that the EPPO standards are not static documents. When information through scientific research or other is developed that can be included in the standard to improve the recommendations of the standard, the panels will consider that and consider an update of the standard. A new standard that has recently, at the end of last year, which was recently adopted, is the PM399 General Guidance for Export Certification Process for Plants, Plant Products, or other regulated articles. So these two standards used in association can be utilized as the overarching information for the more specific standards. When I talk about more specific standards, here we have a list of a number of different standards, and the important factor with this slide is that we have inspection standards for inspection of places of production, and we have inspection standards for inspections of consignments at points of entry. These standards may be, as I've already mentioned, they might be specific to the commodity, or they may be specific to the pest. For this presentation, for the next 4 or 5 slides, I will focus on the 2 inspection standards related to Xylella fastidiosa, so inspection of places of production and inspection of consignments. Of course, it's important, so I've added this slide, it's important to highlight that plants for planting are a high-risk pathway for the entry of Xylella fastidiosa. And importantly, phytosanitary inspections are used to determine the compliance of the consignment or phytosanitary regulations at the place of production based on visual examination for the detection of the pests. In this case, we're going to talk about Xylella in association with documentary checks, etc. I won't read all of this text on the slide, but each of the EPPO standards has a specific scope. The scope details what's in scope, what the standard covers, but it also, where necessary, details what the standard doesn't cover. So just to highlight for the scope for the inspection of consignments, it describes the procedures for inspection of consignments to detect Xylella fastidiosa on host plants. The standard can be applied for import inspection, including sampling and identification. And of course, for the inspection of places of production, it describes the procedures that can be undertaken in the place of production for the inspection of plants. Those inspections may be to satisfy export or for internal country movement or as an element of a national survey. Both standards, inspection of consignments and inspection of places of production, have a similar structure and also similar information. When you're talking about host plants, there's no difference, really. And so within the inspection of consignments, we have the specific scope, as I showed you. We always have an introduction within the standard, but it's not like an introduction for a scientific paper. It's normally much shorter and includes key information of the relevance of the standard. For example, it may include information on the current distribution of Xylella fastidiosa in the EPPO region, potentially the history of occurrences, detections. We also include information on the vectors for Xylella as well. Of course, information on the host plants concerned is included in the standard. And if you look down to almost the bottom, Appendix 1, there's detailed information included in this appendix on the symptoms of Xylella infection on some of the main hosts. So this is really important because symptoms for Xylella can be highly variable, and the expression could depend on the host plant, the strain of the bacteria, environmental conditions, growing conditions of the plant. So whether you're inspecting grapevine, peach, citrus, or olive, it's very important to have detailed guidance on the potential symptoms that you may encounter. And also within the inspection standards, we include images of those symptoms as well. Of course, the standard has a detailed aspect on phytosanitary inspections, and I'm going to look into a little bit more detail of these last bullet points in the next couple of slides. So we have the information on phytosanitary inspections. Remember, when we're talking about phytosanitary inspections, we're talking about the official visual examination of plant products or other regulated articles. But of course, we have detailed guidance for the inspectors on the collection of samples to send to the laboratory, how you would preserve those samples, and potential ways of transporting those samples for laboratory testing. One more thing to note for right at the bottom is we have a short procedure for inspectors, and I think this is a really useful aspect of the inspection standards. Therefore, it enables the inspector to take that 1-page or 2-page shortened version of the inspection standards there to the field with them, or have it on the tablet a little bit, and so it's a little bit more easier to look at. It's important to note that in the inspection of places of production, there is a difference, of course, because we're looking at the place of production. So we would have information on inspection and sampling periods. When is the best time to go to the place of production to have the maximum chance of either finding symptoms or detecting the bacterium? I talked earlier about the importance of research to improve the EPPO standards, and the X Factor Actors project, which is finished for a little bit of time now, but it produced some amazing research on exactly that. So for a number of the host plants of Xylella, the research gave really good guidance of the timings to go, with the timings where samples could be collected for maximum efficiency of detection. Of course, the standards give important information on health and hygiene considerations. It's important that the inspectors use protective shoes, disinfect tools, etc., when they're moving between lots or consignments. Again, we've already talked about the inspection locations and timings. It gives information on lot identification and the how you can look at that, especially on risk-based sampling as well. Inspection and sampling units. Information is given, which the information comes from the ISPM standard number 31 methodologies for sampling of consignments. ISPM 31 also, though, can be used for inspection of places of production. And it gives information on, for Xylella fastidiosa, it is important to have a high confidence and the capacity to detect the bacterium at a low level of infection. Of course, visual observations alone are not sufficient for the detection of Xylella fastidiosa. So it also gives information on sampling, asymptomatic sampling. Symptomatic sampling, of course, should always be carried out. But it gives some really useful information on the number of branches, the number of leaves, the minimum numbers to be sampled and sent to the lab. And again, this information was developed by scientists within the X-Actors project. So that information was included into the inspection standards. I think also Charlotte will look at that a little bit later. I think this is my last slide, but also it includes the inspection standard, includes information on sampling of vectors, of course, whether that's if you find one in a consignment at a point of entry using booters, or whether that's sticky traps placed in the place of production. Finally, the inspection standards gives some information on how to seal, label, and preserve the sample that's collected, whether that's leaf samples or whether that's vectors in ethanol, and how they should be sent to the laboratory immediately. Charlotte.
So thank you, Rob, for this nice introduction about inspection. So of course, once inspectors have collected samples, the next step is to send them to the laboratory so the pest can be detected and identified. And so at EPPO, we also provide diagnostic standards to EPPO, and the objective is to ensure the accurate detection and identification of regulated pests, which is of course essential then to the EPPOs so they can take appropriate phytosanitary measures. So next slide, please. So in the series PM7, we prepare diagnostic protocols for regulated pests. So these are internationally agreed diagnostic protocols, and they describe the tests that are needed for the detection and identification of the pests. So sometimes for that you will only need one test, but most of the time you will need a combination of tests to really be able to say this pest is present in the sample. And the tests that are recommended in EPPO diagnostic protocols, they are selected, and they should preferably meet the following basic criteria. The first one is that the diagnostic laboratories in the EPPO region should have some experience with the test, because sometimes tests are described in publications, for example, but then when you start to routinely apply it for diagnostics in many samples, you realize that they are not appropriate. So experience is very important. And then, of course, the test should be validated, and the performance characteristics of the test should be provided. And usually this information is provided in relevant appendices that we have in our standards and that describe the tests. And also the test, the description, and the validation data should be publicly available. So ideally, they should be published in scientific publication, and at the minimum, they should be available online. So next slide. Yeah, next slide, guys. So our diagnostic protocols are prepared by different specialized panels. So we have 5 specific panels in total, one for bacteriology, one for nematology, one for entomology, one for virology and phytoplasmology, and one for mycology. And all these panels are in charge of preparing and reviewing the specific diagnostic standards in their disciplines. And they work under the supervision of another panel. Next slide, next please, yeah, which is a horizontal panel on diagnostic and quality assurance. And this panel is in charge of preparing horizontal standards. So mostly standards regarding, for example, accreditation or how you will report your results. How do you do to prepare reference material, for examples? And they also have in charge an horizontal standard on how to use the specific standards that are prepared by the specialized panel. And next, yeah, so for Xylella fastidiosa, of course, it's the panel on diagnostic in bacteriology that is in charge of this standard. I put you the link where you can find the composition of the panel, which meets, so the panel meets every 18 months. And as I said, it is in charge of developing and reviewing the specific diagnostic standards in bacteriology. So in total, currently we have 27 specific diagnostic standards, and yeah, you can find a list in EPPO Global Database. If you want to look at it, I put you the link. Next. So regarding Xylella fastidiosa, the standard was first approved in 2004. So it's actually an old standard. And after the different outbreaks that appeared in the EPPO region, it has been revised. So 4 times in 2016, 18, 19, and 23. So we are at the 5th version of these diagnostic standards. And of course, at each revision, the output of different EU-funded projects have been taken into account. So for example, X-Factor, Ponte, and also results of, for example, Ufrexscope projects. So next. And also, of course, next, yeah, the experience and the validation data that were gathered in EPPO diagnostic laboratories was also taken into account. So it's not always published or part as a research project, but it's really an important work that is performed by diagnostic laboratories, in particular now that they have to be accredited for performing official diagnostics. There is a huge work that is done on test validation in all the labs. And of course, this experience is also important for our diagnostic standard and for doing the recommendation. So next slide. So the diagnostic standards are structured always the same way. So as the inspection standards, you have an introduction that will provide some information regarding the history of the pest and also where it is from. And we try to keep it short and to focus on information that are of diagnostic importance. So yeah, it depends on the pest, and it also depends on what information is available in other EPPO documents. We also then have a section on identity, where we provide the name of the pest and also the synonyms, information on the categorization of the pests. Then we have a section on detection. So this section focuses on the tests that are used to show the presence, the first sign of presence of a pest and it is separated in different sections. So the first section is disease symptoms. So Rob already provided information because this information is also an inspection standard because it's important for both the inspectors, but also the people working in the lab because they have to be able to check for the presence of the symptoms on the material they received. And also in some NPPOs, actually, people from the labs are also going in the field. So it's also important for them to know the symptoms. So we have information on disease symptoms on different host plants, the most important one, and for the most common diseases that are observed. Next. Then we have information for sampling and sample preparation in the laboratory. So it's a little bit different than for the inspection standard, because it's really about you receive a sample and what you will take from this sample to prepare the plant material that you will need for your analysis in the lab. And we describe this sample preparation both for plant material, but also for vectors. And this is, for example, a table that is present in our standard, where we give information for different host plants, the minimum of leaves and twigs that needs to be collected for plants by that needs to be collected per plant, and also the number of plants that can be pulled in a single analysis, and how to process the tissue, and the volume of extraction buffer that you need to add in each different cases. Next, please. So then we have different section for detection still. So we describe the different screening tests that are available. In Xylella, there is also a section on additional tests and also a section on isolation. So I will come back on this, but in the case of Xylella, compared to other standards, isolation is actually not recommended for detection, because it's quite difficult to isolate the bacteria. And so detection is more focused on serological and molecular tests. But then if you want to further identify, it's still important to perform isolation and then to try to identify your culture. So section 4 is about identification and subspecies determination. So you have a section on identification of pure culture for Xylella fastidiosa, and then you also have a section on the molecular tests that allows the identification and also the assignment of Xylella fastidiosa subspecies. And the last section is on the pathogenicity test. And then we have some sections that are common to all our standards. We provide information on where to find reference material, how you should report and document your results. And here you have a cross-reference with another horizontal standard that is specifically on that point. We refer in the performance characteristics session to our database on diagnostic expertise, where you can find additional validation data for different molecular tests. And then we also provide some contacts points so experts of Xylella fastidiosa that you can contact if you have further information on the standard. So next, Rob. So with diagnostic standards, we usually provide flow diagrams, which is kind of a summary of the diagnostic process and of the tests that need to be performed in order to detect and identify the pests. So in the case of Xylella fastidiosa, as you can see, you first have to perform for plant material screening tests. And if your tests are negative, you will be able to conclude that Xylella fastidiosa is not detected. But if the tests are positive, you need at least 2 positive tests, you will conclude that Xylella fastidiosa is detected. And as I said before, Isolation is kind of recommended afterwards, but it might be difficult. So you have also the option, if isolation is negative, to attempt subspecies assignation, using molecular tests directly on the plant extract. But if your isolation is positive, then you can perform your molecular test or your serological test directly on this, and you can identify Xylella fastidiosa in culture and then assign, if you can, the subspecies. And we also have a flow diagrams for vectors, which is a little bit different than for plant samples, since you will only have a direct screening test on the DNA extracted from the vector. And here as well, you need at least 2 positive tests to confirm that Xylella fastidiosa is detected. And you can also attempt a subspecies assignment by molecular tests. Next, please. Yeah, you can go in series, Rob. So I just put this to highlight that all the tests that are recommended in EPPO standard are then described in details in appendices. And in these appendices, you will have general information on the test, so on the scope, how the test was developed and by whom, and also providing information on the primers. Then there is the method that is described, how you do perform nucleic acid extraction and purification, detailed description of the master mix that should be used and on the PCR conditions. Then section 3 is always about essential procedural information, so the controls that you need to use to ensure that your test results are appropriate. And also in section 3.2, how you interpret your results. And then section 4 is always about the performance characteristics available for the test. So it's just an example of the conventional, one of the conventional PCR that is described in the standard, but it's usually the same structure for all the molecular tests. So you can really quickly identify the information that are of interest. Next slide, Bob, please. And yeah, this last slide was just to mention that recently there has been a new ISPM 27. So the ISPM, or the international standard that are prepared at by IPPC, so at the International Plant Protection Convention, which is for the whole world, so not just for the EPPO region. And this standard was drafted also by experts that were involved in the preparation of the EPPO standard, and it really takes many of the elements from our standard. So just to highlight the fact that EPPO standards, they are not just for the EPPO region, and they are really recognized also at international level. And they are often used to draft ISPM standards. Next. And one last slide to highlight that at EPPO, we also maintain a database to support plant-based diagnostic activities that contains sequences that have been curated. And for Xylella fastidiosa, using the 16S rDNA and the MUTES barcodes, you can identify Xylella fastidiosa at the subspecies level. So on that website, you can find protocols and also reference sequences that would allow you to accurately identify the subspecies using sequencing. So for the last part of the talk, I just would like to highlight one of the important missions that we have at EPPO, which is to disseminate the information that we gather by and that member country governments and experts gather on outbreak, spread of pests, and also any important information regarding pests that are provided by experts. And for that, we establish documentation and information service, and we publish all the information in different databases and also documents. So just to mention that our most important database, I think at EPPO level, next slide please, is EPPO Global Database. It's really a node where you can find most of the information that is gathered. So for example, for Xylella fastidiosa, you can find information on the distribution, on the host, and now for the host, if you click on the little information button, you will also have information on where the information comes from, the publication that identify that this species is a host. We provide information on categorization. There is on vectors, and also there are many pictures that are provided in EPPO Global Database. So we highlighted how symptoms are important and how recognizing symptoms is important. And for that, it's really important to have pictures to compare. So any expert can submit pictures on EPPO Global Database, and we really invite them to do so, because the more pictures we have, the more information and the more information is shared between experts. Next slide. So in EPPO Links, which is a kind of new substructure in EPPO Global Database, you can find information also that we have on Xylella fastidiosa in our other platform. So the first one is the platform on communication material, where NPPOs can share all the communication campaign they are doing on a specific pest. And you can also find information that is published in the EPPO PRA platform, which gathers all the pest risk analysis that are performed by EPPO, but also in any other countries or region of the world. And so yes, EPPO Global Database is really a node. And lastly, you can also find information on the reporting service that we publish. So every month we publish information on new pests that have been found, or update of presence of pests in the world. And all this information are gathered in what we call the EPPO reporting service. And this information is also available in EPPO Global Database. And then finally, just to note that all our standards are published in our journal, the EPPO Bulletin. And at least for PM3 and PM7 standards, they are all freely available, so you can access them. So on this, I would like to thank you all for your attention. And yeah, I think we can take a few questions if you have.
Okay, I think I will help you with the questions in blank. I think I had to leave. So thank you very much, Rob and Charlotte, for excellent presentation.